The question is, which of the following is the final electron acceptor in aerobic cellular respiration? Here the two terms you have to observe is final electron acceptor and aerobic cellular respiration. What is aerobic cellular respiration? Means the cellular respiration occurs at the presence of oxygen. Final electron acceptor means that is oxidative phosphorylation. In the cellular respiration, in aerobic conditions, there are three stages are there: glycolysis, Krebs cycle, and electron transport chain. In the glycolysis take place in the cytoplasm, and the Krebs cycle take place in the mitochondria matrix, and the electron transport chain take place in the cristae, the primary particles. During the glycolysis, the glucose is degraded partially. Final product is pyruvate or ethyl alcohol, whatever it may be. But glycolysis does not utilize oxygen. And in the Krebs cycle, the pyruvate or ethyl alcohol enter into the Krebs cycle that can undergo the further degradation and produces the carbon dioxide. This Krebs cycle also does not use the oxygen. And the final one is the electron transport chain. In the electron transport chain, the oxygen is utilized as the high electronegative molecule, the high oxidative molecule, which can be reduced and form the water molecule by grabbing the electrons from the electron transport chain and liberate the energy. That energy is fixed in the ATP molecule. So here the pyruvate is forming during the glycolysis process. So here the answer is not correct, the pyruvate. And the carbon dioxide is formed during the Krebs cycle, that does not accept any electrons or take electrons. Hydrogen ion, that hydrogen ions are pumped into the intermitochondrial membrane in the electron transport chain, but that is not meant for the accepting electrons. So the answer is oxygen. But we have to go for the details. So we have to elaborate our discussion about the cellular respiration. In the cellular respiration, we are concentrating on the oxidative phosphorylation. What is phosphorylation? It is nothing but ATP synthesis is the phosphorylation at the presence of high oxidizing agent, that is oxygen. That's why it is called as the oxidative phosphorylation. Here the oxygen is involving, which is high oxidizing agent state. Oxidizing state. The electronegativity of this molecule is very high, that is 800 millivolts high electronegativity. That's why it is grabbing the electrons from the electron transport chain. That is we want actually. The cellular respiration required to grab the electrons from the system. If the electron, what is the need to transport the electrons or grabbing the electrons? Means electron transportation can liberate the energy. That energy is fixed in the ATP. That is called oxidative phosphorylation. Actually this is third stage in the cellular respiration. In the first stage, that is glycolysis. Glyco means glucose, the 6-carbon compound is degraded into 2 pyruvate molecules which contain 3 carbon. Each one contains 3 carbons. This is 6-carbon compound. It can go through the 10 series of reactions and finally degraded into 3 carbon compounds called the pyruvates. At this time, in this glycolysis, the glucose is partially degraded and form ATP molecules, and high amount of energy is fixed in NADH. This is we are actually going to discuss. Number 2 stage is Krebs cycle. In the Krebs cycle, which is taking place in the mitochondria matrix, means the NADH molecules which are formed in the cytoplasm. This is actually take place in the cytoplasm. From the cytoplasm, these NADH molecules enter into the mitochondria matrix. Also, these pyruvate molecules are also entered into the Krebs cycle. These pyruvate molecules can go through the series of reactions, cyclical reactions in the Krebs cycle and forms GTP molecules, which can be converted into ATP molecules, and the most amount of energy is fixed in NAD and FAD molecules, FADH2. Now these are actually oxidative state and these are reduced stage. Means the NAD are oxidized state, FAD also. Now these are reduced stage. Now these NAD molecules, NADH molecules formed in the cytoplasm and the NADH and FADH molecules of the Krebs cycle are present in the mitochondria matrix. Now the third stage, oxidative phosphorylation, which is taking place in the primary particles of cristae located in the inner mitochondrial membrane. This is outer membrane and this is inner membrane of mitochondria. These foldings are called the cristae. In the cristae, the primary particles are present. In these primary particles, actually, the oxidative phosphorylation or electron transport chain is taking place, etc. Now, the NADH molecules formed in the glycolysis and in the Krebs cycle are ready in the mitochondria matrix, FADH2 molecules. Next, the primary particles or inner mitochondrial membrane is containing some enzyme complexes which can transport the electrons. These are the enzyme complexes, first, second, third and fourth. There are some names are there, cytochrome Q and other electron transporters. These four are electron transporters, and the final electron transporter, fifth one is ATP synthase complex. This is ATP synthase complex. Okay. Now these are in reduced state. Oxygen is available in the mitochondrial matrix. That oxygen is oxidized state. Now what is happening? These reduced NADH and FADH molecules release the electrons. They can transport it to the different electron transporters and finally accepted or grabbed, received by the oxygen, because it is high oxidizing agent and electronegative molecule. Then the ATP is synthesized. We will discuss how is it. NADH can be divided as the NAD plus release the protons and electrons. FAD divided as the protons and release the electrons. These electrons are accepted and taken by these different electron transporters and they can be transported. So here the electrons are transporting. While the electrons are transporting in the universe, the energy is liberated while the electrons are transported. Here in this case also, by the electron transporters, the electrons are moving or transporting, then the energy is liberated. Where is that energy? That energy is used to pump the protons into the intermitochondrial space. This is intermitochondrial space. This intermitochondrial space is accumulated by the protons. These protons are actually pumped by the energy which is liberating from the electrons. Means the energy which is liberating from the transportation of electrons is not wasted. That is used to pump the protons from the matrix into the intermitochondrial membrane against the concentration gradient. Now, after some time, this intermitochondrial space is filled with the protons. highly packed with the protons. The highly packed protons are containing some energy, that is called the potential energy. Now, this ATP synthase complex can pump the protons into the mitochondrial matrix, repump the protons into the mitochondrial matrix. The potential energy present in the protons is converting as a kinetic energy while the protons are moving into the mitochondrial matrix. Then what happens in the mitochondrial matrix? These protons combine with electrons and takes one oxygen atom and formed as H2O. Here I can write the equation here. Here I can write H plus 2H plus 2 electrons plus 1 by 2 O2. 1 by 2 O2 means these two will be cancelled. Then what happens? It can form one water molecule. Here what is happening? The high oxidizing agent, the oxygen, is grabbing the electrons, pulling the electrons from the electron transport chain towards it and formed as the water molecules. This electron transport chain is very important to liberate the energy. Why? To pump the protons into the intermitochondrial space. Why is it pumping the protons into the intermitochondrial membrane? To pack the protons. Why is it packing the protons? To get the potential energy. Then what happens? This potential energy can be converted into kinetic energy while it is passing through the ATP synthase complex into the mitochondrial matrix. Finally, the oxygen is accepting the electrons and forming the water molecule. So this is called the oxidative phosphorylation. During the oxidative phosphorylation, the electrons are finally accepted by the oxygen, high electronegative molecule, and forming as a water molecule. This total process is actually running by the oxygen. That is why it is called as a oxidative phosphorylation. In the anaerobic respiration, total amount of energy present in this glucose molecule or hexose sugar is liberated. That is approximately 36 ATP molecules are synthesized, are formed after completion of aerobic respiration. But in the anaerobic respiration, only the hexose sugar is partially degraded. Few ATP molecules are synthesized. In the aerobic respiration, the hexose sugar is completely degraded in the 3 stages and converted into synthesize the ATP molecules finally. This is called the oxidative phosphorylation. The final electron acceptor is the oxygen, but not the pyruvate or carbon dioxide. Here, during this Krebs cycle, the carbon dioxide are formed. But carbon dioxide do not grab any electrons, do not pump any protons. So the pyruvate is not pumping the electrons or not grabbing the electrons. So the carbon dioxide is also not pumping, and H plus ions also not taking the electrons. So the final electron acceptor is oxygen.